Welcome to our presentation today. My name is Andreas Scherer. I'm the CEO of Golden Helix. With me is Gabe Rudy, who is in charge of product development at our company. He will be the main presenter for this webcast today. On behalf of Golden Helix, we'd like to welcome you to our webinar. Today is a big day for us. First and foremost, we got 2 feet of snow here in Bozeman, Montana, and that means that half of the city is probably on the mountain by now. But not us, because we have exciting news to share. We're going to launch a major release of our flagship product SVS to the general public. SVS 8 is a substantial improvement over the previous release in a number of dimensions. There is almost too much to go through in terms of improvements within the next hour, so we will try to hit as much as possible the highlights. This new product release is very significant, and we came a long way. Over 5 years ago, in November 2008, we launched SVS 7 as a powerful tool to conduct next-gen sequencing and GWAS studies. Since then, we have been adopted by hundreds of client organizations worldwide. SVS is used by leading research organizations in the US, Canada, Latin America, Asia, Australia, Africa, and of course Europe. About 1.5 years ago, we launched the first version of Genome Browse. This tool was designed to help researchers to view large sequencing files alongside with public annotation databases in a fluid and intuitive way. Thousands of researchers in our field are using Genome Browse as of today. Now, as our customers have seen the intuitive way to navigate through extensive databases and large amounts of data leveraging Genome Browse, we have been asked to bring the same look and feel to SVS. And this becomes increasingly important as SVS is used outside of our core group of users. Besides researchers who analyze the human genome, respectively plant and animal DNA, our software is also used in clinical settings where translational scientists are working hand in hand with their clinical counterparts to use genomic information, diagnosing diseases, and identifying best treatment options for patients. Also, we see now more and more a third group of users coming up, and that group becomes increasingly more important to us, and those are undergrads and graduate students. They learn with SVS how to conduct work in their field for the first time. Here, the ease of use of our platform reduces the initial hurdle to enter this specific field. It eliminates the need for a student to become a computer science whiz first before they even can begin to analyze data and focus on the science behind it. Throughout this webcast, you will hear about references on how we improve the usability of SVS for that reason. Another major topic that we addressed in this release is scalability. As sequencing technology has become cheaper, there's more and more data waiting to be analyzed. Sample sizes of studies are increasing, and our idea of SVS is that we put a big punch in a small package. Our users can conduct most of their work leveraging small servers or even their desktop. This means we have to work hard to make the ins and outs of SVS as scalable and as efficient as possible. Over the last few years, we reinvented core capabilities of SVS. For example, we substantially improved how we represent and manage data, simplify the import of data, and improved all kinds of data manipulation capabilities. Again, Gabe will focus a lot in his portion on specific examples of that. As we have improved SVS and its global adoption increased, our company has matured. We gained a lot of process knowledge in doing substantial service engagements for our clients, primarily in the pharmaceutical and bioscience space. We became an active participant in the community by speaking at conferences, sponsoring key events, as well as facilitating this monthly webcast series with a global audience. Our website became over the years a knowledge hub for our user base to learn about recent developments, but also best practices in our field. In many ways, this launch is indicative of the level of maturity that we as a company have reached. I took some time over the weekend to summarize my thoughts in my blog post that you see here on the right. So please feel free to take a look when you have a chance. It's now my distinct pleasure to hand this meeting over to Gabe Rudy, who is very excited to present the new capabilities of SVS8. Gabe?
Thank you, Andreas. I'd like to jump right into covering the agenda of this webcast, but before I do that, I want to touch on some important sort of housekeeping points. One will be that we'll save some time at the end of this webinar to go through any questions you might have. You don't have to wait till the end to enter those questions, though. On GoToMeeting, there's a little questions dialogue in your panel. You can go ahead and ask your questions at any time, and then we'll take them at the end. And one of the most commonly asked questions I can answer now is, yes, we will record this webinar and the slides will be available. And in fact, Jessica has already posted the slides through the GoToMeeting chat interface if you want to see those. So don't feel free like you have to take desperate notes or anything, but we will cover a lot of great ground. Okay, so to cover really what is in SVS8, I feel like I have to spend a little bit of time establishing a bit of the historical context of how we got here. And this is important because as a major release, going from 7 to 8, this really is a release where we focused on infrastructure, about things we've always wanted to do, but it took a lot of, essentially we needed a time in which we could fully invest into building a large infrastructure to make it happen, and that's what we're doing in this release. And I'll touch on how that new infrastructure is useful in SVS and Genome Browse, as well as how Genome Browse itself as an individual technology has matured. Even though we'll be talking a lot about the visualization and annotation capabilities of Genome Browse and SVS, that expansive experience and visualization will enable any type of analysis in SVS to be more powerful and useful. So if you're doing genome-wide association studies, which is still a strong and active part of our user base, you'll be able to take advantage of this technology. But we're not standing still either. We have continued to add methods to our statistical capabilities of SVS, and I'll finish up on that. So I'm not going to go into too much of a background into Golden Helix. We've been around for 14 years, and I assume most of you have had some experience with us, either as a user or just seeing us out in the community. We are based in Bozeman, Montana, and as Andreas mentioned, we just got dumped on last night. This is actually a picture of my car this morning. We barely made it to the office. That is not a small car, actually. That's a full-size SUV. There's about 3 feet of snow in my driveway, and I had a hike on down to the plowed road where Andreas came and picked me up. So we made it here one way or another, and so excited to be presenting about our two products, essentially, SVS and Genome Browse, on how they're coming together today. So let's touch on that historical context. Like I said, one of the themes of this webcast will be an investment in infrastructure and how that infrastructure is empowering to be able to do work that would potentially not be able to be done on a desktop or would take an extreme amount of bioinformatic expertise. And this is not a new theme for us as a company. I'm going to point out the fact that back in 2007, where we had Helix Tree 6 and then we went to Helix Tree 7, that major release in February 2009 was also a massive upgrade in our infrastructure. We completely rewrote how we handle spreadsheet data sets so that we could easily scale to whole genome scale analysis. And it's been actually serving us very well. Our still core SVS infrastructure is being used today by people who are doing exome projects on studies with potentially tens of thousands of samples and whole genome projects with thousands of samples, etc. But within a couple months of getting our SVS 7 release out, we knew we needed to start working on the visualization component, because all that statistical power is really only the starting place, and the real insights come when you are able to visualize that data. So in 7.3, we released our first genomic scale visualization, where we will where we plotted the analysis results from our statistical work in a genomic context, and we were bringing in public data sources, etc. Of course, we didn't stop there. We continued to expand as the world got very focused on next generation sequencing, and it became a core driver of our users' research. We built out small N analysis workflows such as trio-based analysis, as well as large N analysis workflows such as collapsing methods, also built in RNA-seq as well as DNA-seq capabilities, and even bolstering our statistical methods to keep up with the times. In our last public release, we released mixed models. But even back in 2010, after we had gotten our first release of our genomic-based visualization and SVS out, any complex system, when you build it as an engineer, you immediately realize that after you build it the first time, you know you could do a better job building it the second time. So within a year, we had already started placing on our whiteboard our ideas of how our user experience could be elevated with a completely rebuilt visualization, a genomic visualization tool. Well, we also knew that our infrastructure needed to be revamped as well to be able to handle the 10 gigabyte, 100 gigabyte BAM files, the alignment data, the variant data, all these different file formats. And so that we went to work on that. And within a year, we were shipping our first version of Genome Browse. We had people comment that this was a very polished and very, they were surprised at how polished and beautiful this interface was, and it really was because it was our second pass at this. It was our complete rewrite essentially of what we had in SVS, and we continued to add support for new file formats, direct reading of VCF, etc. But it took us a while to essentially to build out the features that we had accumulated in our SVS visualization, as well as in SVS 8, and I'm going to touch on this next, we decided that there was another big leap of infrastructure to be done. And so that is what we are talking about today, is the merging of these two great product lines. And I'll remind you that Genome Browse as a standalone product is a free tool, it's a visualization tool, and we'll be talking about the release of SVS 8 today, and shortly on its heels will be the release of Genome Browse 2.0, continuing its path as a free tool, a standalone tool. But everything that we'll be seeing today is essentially that same core technology. Okay, so what is this infrastructure that I'm talking about that we invested in heavily to bring into SVS 8? Really, there's four parts of it. The first part is that we have built from the ground up a completely new file format. As I mentioned, SVS 7 came out with a new tabularization engine so that we could handle tabular data with millions of columns, tens of millions of columns, hundreds of millions of columns. And when we built our visualization engine, we built a new file format to handle genomic scale data that had genomic coordinates. This file format is essentially the best of both worlds plus more, and I'll touch on that. But one of the things we also realized is that with all the public data sources out there, the bottleneck was getting those into a usable form. And we completely rethought and rebuilt how we import data or convert public data from various different file formats into our into our system, into our new file format. Now, all that public data isn't useful until you can actually apply it to your own samples and you're annotating your own data, and we've updated our annotation workflows to make that much easier. And finally, we've been doing this for so long that we realized that there's no way to keep pace with sort of the releases of new data without having an extremely well-structured, ordered, and versioned repository system, and we've redesigned the way we handle our public repository so that it's very easy to keep up with the latest versions of data. So, what is this file format that I keep on mentioning? Well, I'm calling it TSF, Tabular Storage Format, and I wrote a lengthy blog post back in December, when I was in the middle of implementing it. And really, like I said, this has been something that we have been designing and redesigning for years, and now is the opportunity to invest in making this leap into this new technology. What's great about it is a couple things. One is that it's built to scale. I'm already just, if people in the office might have seen me skipping around the last couple months, it's because it's really worked out the way we expected it to, and that we're able to throw new versions of these large data sets, things like 1,000 genomes, the database of non-synonymous functional predictions, and often get the the file format itself is a 10-to-1 compression ratio, where the file is in a compressed format, but it's also in a usable format. And I'll show you some of this a little more interactively as we go through the demonstration format. The next thing I mentioned is that getting your data into this format is and has been potentially a bottleneck. Getting your data into any usable format for any analysis is always a bottleneck, but we have found that people were often having to write one-off scripts or just rely on our own support staff to do this type of conversion work whenever someone had a custom file that they needed to convert and use. That's fine, our support staff is there to help, but we realized that it would be more empowering if we could build an interface that handled the myriad number of file formats that are out in the wild that people want to use. And more importantly, you needed a way to actually see what was going on through each step of the conversion process. So our new Convert Wizard was built from the ground up to be extremely interactive, with lots of previews of the data you're looking at. As you change a setting, you immediately get a preview what that setting changes. You know, if you change the file delimiter, you get to see what that looks like, etc. And it's intended to be extremely self-explanatory. We have little help messages as we go. Finally, the whole point of this is that once you get to your new imported data, you can use it for your analysis or your visualization. And the great thing about this format is it's a single file that you can throw up on a Dropbox folder or email to a friend. And as I mentioned, it's already in a very compact format. Okay, so we have a great repository of public data sets and maybe some that are private that you have curated yourself. One thing we realized in our SVS analysis workflow was that people were often having to go through a set of steps to get to their final filtered and annotated results. So with SVS 8, we allow you to take a number of annotation sources. You know, in this case, we got OMIM and RefSeq genes, and a couple variant catalogs, and you can select all of those up front, and then we guide you through some options on each one. Do you want to annotate against this or filter? Do you want to pick a threshold, etc.? And then boom, we sort of set up a chain of filters to do that all for you at once. This saves you time, but it also just makes it easier to do analysis that you already know what you want to do up front. Of course, if you want to still do things one data source at a time, you can do that, but this is something that we're quite excited about having to help improve the efficiency of that analysis. Now, as I mentioned, we went through the effort of rethinking how we're going to build a data repository that can really keep up with the paces. So one of the things about this is that we have essentially built into the file format a very rich set of documentation fields. You can see information about how we curated the data source, if we did any sort of manipulation or changes. You can see information about where we downloaded it, the citation of the paper, if there was a paper involved with the creation of it, as well as one of the things that's very useful in the context of data sources that change fairly rapidly, like the 1,000 genomes or dbSNP, is that we added a very simple versioning system. So we have a series name, and for something like dbSNP, you will by default only see the latest version of that annotation track, instead of every the 5 or 10 versions that we have curated. But here's the important thing. This is something that, although we'll throw the latest version up there as soon as it's available, you don't have to upgrade to it. If you're in the middle of a project, it's just as easy to continue to work on your older version, and we never remove it from our servers. So if you're linking to dbSNP 137, continue to use that for the end of your project, and at your own convenience, you can upgrade to dbSNP 138. Anything that you've downloaded, of course, you can continue to use, but our server will never remove historical versions of data sets, and you can always rely on those being there for reference. Okay, let's go see what some of this looks like by pulling up SVS here. So I'm going to go ahead and minimize my PowerPoint and pull up SVS. So if you're familiar with SVS, this is our start screen. It's a little bit redesigned for SVS 8, but all in all, it's still the same SVS you'd come to know and love. So I'm going to go ahead and start with a project here that's already had some analysis done. The analysis workflows are more or less the same. We have had the chance to do a restructuring of our menu system to allow for a more intuitive workflow-based organization, and we'll use some of these menus later on as I touch on some of these analysis things, such as that new annotate and filter capability. But here I have a spreadsheet in which I've already filtered down a trio, so 3 samples, down to 3,000 rare variants using our public annotation sources. Now I'm going to go ahead and plot this data in Genome Browse. I'm going to plot this variance as a variant map. And now we're using that same Genome Browse engine that you might have come to know and love as well to do the actual plotting of project data. And of course, alongside your project data, such as these 3 samples, can be public data sources such as the latest RefSeq genes from curated from UCSC, but RefSeq, of course, is a database maintained by NCBI. And of course, you have that interactive fluid interface where you can click around, expand, and zoom on in. And we're looking at a density rug here. And as we zoom in, we get to see the individual variants and their individual genotypes for a sample. In this case, I'm just looking at a regular homozygous variant. Maybe we can find some other information here. So here's a sample where one of the parents has homozygous and the other 2 do not have any variants, and you can see which gene it's in, etc. So let's go ahead and add now to this some public data. And to do that, I'm going to go to our new Add dialog. I'm going to touch on this a little bit more later, but this is our completely newly designed interface for managing data sources. And as you can see, it's very easy to look through our public annotations, and you saw a screenshot of this earlier, but this is now a new organization where you can look at all public annotations and search for something you're looking for, like dbSNP, or you can go down to a different folder and just find, for example, the dbSNP that is in the variant and function versus, say, the gene and regulation section. Now, this is a preview of our updated server, which will contain the latest version of dbSNP. It also contains the latest version of, say, the database of non-synonymous functional predictions. This is our largest database because it contains a single set of data for every single possible missense mutation or DNA change in a coding region of the genome. So if I uncheck this latest box, you'll see that this database has had a number of revisions going back to version 2.0, and now the latest version curated just last month is version 2.3. But by default, we're just looking at that latest version 2.3. And the other thing we can start to see is this documentation that's built right in. And when you click on any data source, you get to see information about the data source, information about how many variants or features are in there. In this case, there's 90 million. That's right, almost 100 million variants in this database. The file is only a modest 1.5 gigabytes, and we'll see more about that in a second. And you can actually start to see the information about the fields, or the, essentially the columns in this data set, as well as their documentation. So this is a data set that contains functional predictions, SIFT, polyphen, mutation tester, Fathom, for every single non-synonymous change in the human genome, all 90 million of them. So we actually are storing both the categorical prediction of the sort of the enumerated of whether something is a disease causing, as well as the actual continuous score that the algorithm produced. Of course, we have that documentation, information about where this data source came from, the citations of the papers, and then this is something we've added with our new TSF file format. As I mentioned, this is a format that uses a internal compression. Because it's storing data essentially on a column basis, we can give you compression on a per column level. So you can see some fields are extremely compressed down to 5% ratio, where the raw data was 300 megabytes and the compressed data was 18 megabytes. All in all, you can basically see that this file, if you were to print it out to a text stream, would take about 12 gigabytes of data, and that's actually the size of the raw data source that you would download from their website. Yet in its usable and very highly annotated form here, curated form, it's just a mere 1 gigabyte that you can download onto your hard drive, or you can use it directly from our servers. And that's a new feature in this release as well, is if you have a small data set and you don't want to download something like this, it's very easy to just annotate against our remote network-based data sources. So let's look at dbSNP 138 as well. We can see that it's got a number of new fields that weren't there in the previous releases, and information about its curation, about a 15% compression ratio. These numeric fields are a little more data dense, so a little bit less compression ratio than all those string and categorical fields. Let's go ahead and plot this so you can see that right here in the data in our project, we're now plotting a remote network-based data source and having that same fluid interactive experience as I zoom around in here. One thing I didn't mention yet is that not only does this new format allow you to annotate, plot, even look at it in a tabular sense. I can pull up a table. We do also embed hyperlinks to as many fields as possible. So with RSIDs, you can click on out and open that RSID in DBSNP. And I can dock this table if I want to just keep it handy to kind of navigate through. It's easy to dock it over there. Go ahead and dock it back here. And the other thing is that this field, being a very useful field to look up, can actually be used to search for your data. This is also a new feature of our new file format is you can optionally add fields to an index list, which allows them to be searched. Now remember, DBSNP being as large as it is, it actually contains, if I click on the source itself, we can get that same documentation interactively here as opposed to just in the data source library. It contains 61 million variants in its latest 138 release. So again, quite a large data source. Those 61 billion variants, if you were to try to search for a single RSID in any traditional sense without a full-blown server-side database, would take quite a bit of effort. But with TSF, we've indexed that field, and now I can just say RS12345 and immediately pull up all of the RSIDs that start with that string. And I can go ahead and jump to one of those if I wanted to. I can go find RS12345. I could change the label on my plots if I wanted to make it more obvious. Let's change our label to the identifier. Which one is RS12345? There it is. I can click on it and pull up its information about it. It has information about its submission, and go ahead and click on out to it, etc. So this is without installing either locally or remotely, an advanced database. This is built into the file format, the ability to quickly search for individual features. This works for gene tracks. If I'm searching for a gene, I immediately find that gene. If I'm searching for an RSID and DBSNP, etc. Again, something that's almost, you don't think about it too much, but these types of RSIDs are very useful to jump to, especially when you're looking at a paper and want to go find that position in your own data. Okay, so I mentioned how we can look at our public data, and we do have quite an extensive repository, and we work hard to keep that up to date. What about if you were wanting to add a data source that we didn't potentially have into your analysis? And again, this is what I mentioned, something we see very commonly, and before now, something that often required a little bit of manual, command line work. And this is pretty much true for any type of scenario. If you're building, if you're trying to annotate something in Excel, you probably have to take any of these files and write a little program to parse them. The public data sources themselves can be not exactly in a usable form right off the bat. But let's go ahead and go down this exercise of finding a data source that we want to use for our own analysis. So to do that, I'm actually going to go to UCSC, being one of the largest repositories of bioinformatic genomic annotations, and they have this table browser at the top of their page that allows you to very quickly find essentially raw dumps of the tables or the database tables that are backing their visualizations. So for example, let's say I wanted to look at some regulation elements and annotate my data against DNA hypersensitivity sites. So these DNA clusters here, this is actually ENCODE data sort of aggregated. So to do this, you can sort of specify a file name, and let's go ahead and get it compressed, and go ahead and start the download there, get output, just save that compressed file. While that's downloading, and it's going pretty quick, let's click on Describe Table Schema. This gives you a little documentation from UCSC's part about what you're getting yourself into. Now, this is a data source that has about 1.3 million rows, so 1.3 million features essentially. And they have, this indexed field as well as a chromosome start stop. That's good. That means we can use it in genomic space. And it has some name and a score. And if you read down here, basically this is a computational aggregation of many of the ENCODE data sets, the uniform DNase hypersensitivity sites. And essentially these are now the filtered set of sections where you have a cluster of DNA hypersensitivity, and so that could be useful from a regulatory perspective. So now that we have that downloaded file, we'll go back into our library here and hit this convert button. Now this is that interactive wizard I mentioned. Again, we went through very many iterations of the design to make this as intuitive as possible. We have a little context-sensitive help. Let's go ahead and go to Add, go to My Downloads, select that file. Notice it's a compressed file, but we're going to read it directly anyway. And immediately we're giving you a preview of the data. There's a little advanced options toggle if you need to specify subdelimiters or anything like that. I don't think I need to. We also automatically detect and set green the chromosome field, the start field, etc. If we didn't automatically detect it, you could specify which fields are the chromosome and start and stop. And then in a few seconds, we can scan through that data, and then we're doing a couple things. We're detecting the genomic coordinate range. That will help us auto-detect what genome species this data should be referencing. The other thing we're doing is detecting the type of data we have in each column. So just like that description of the table up here, it's correctly detecting that this is a bin field. It thinks that this name field is an integer. Let's go ahead and change that to a string. It looks like even though it's supposed to be an identifier, it happens to just all be numeric data. But this is our cluster name. And as I change the name field here, it automatically updates. And then we'll keep this as our score field. Give that a capital score. And I'm going to drop this bin field. This is just a field used by UCSC's internal database mechanics. Okay, so I'm going to go ahead, and this is the data I want to import. And again, as I mentioned, it detected that this is a human genome data, as well as it turns green here every single chromosome I have data on. So it doesn't contain any mitochondrial data, but it automatically detects, and it also cleans up the chromosome name here. You can see the CHR mapping gets removed, and so we get a nice clean imported data set, and it's assigned to the right genome assembly for us. And here's that documentation I mentioned. We can actually say things about this field, and I can go ahead and copy in maybe the description from UCSC if I wanted to. I could put information about where I downloaded it, etc. And like I mentioned, we could add more fields to help have this be a versioned series like dbSNP. In this case, we're going to go ahead and just continue with this. And now it's ready to import. Now there's some other options I could set, and you would see that if I had advanced options, but at the end of the day, everything should be automatically set up for me to go ahead and convert. And very quickly, I'm turning this raw text file, and remember it was compressed, I don't even have to decompress it to start with, with 1.2 million features into a usable genomic annotation source. So what does that look like? Well, I hit finish. And immediately, if I look at my own user annotations, which is the default location, there is this DNase hypersensitivity site, and it contains information about the data, and it compressed down pretty well, and I can go ahead and plot this. And there's another feature here in Genome Browse 8, is I can plot it in any number of ways. I can choose to plot the interval of just where this data is. But that score field is the interesting thing, and I can plot that as a value. So let's go ahead and plot that in my plot here. And so here it is, and I can actually see where these values are. The one thing to note is because these are sections of or intervals, these data points actually have width. They're not all uniform. These are based on the clusters detected. So although we're looking at numeric data and you can start to see where they are, they actually have width. And so as we annotate these against our own features from our own samples here, we can see which variants in our samples overlap these DNase hypersensitivity site. This mutation does not seem to overlap it, and we can do that more systematically. Before I do that, I want to do one more data curation exercise. I noticed yesterday, or actually last week on Twitter, that ClinVar mentioned that they had a new release. So ClinVar is a database of genomic annotations of essentially clinical significance. So annotating which variants have been classified in a clinical setting by a lab. And if you go to ClinVar's internal FTP site, this is a standard FTP site. It's a bunch of raw dumps. They have a VCF section. Now, VCF being a standard format for representing genomic data, we support curating your own annotation sources directly from VCF. So sure enough, here's a release that just came out earlier this month of ClinVar. Let's go ahead and download that. So ClinVar 2014-03, and let's see what importing that set of variants looks like through our conversion process. Go to Add, click on Convert. Now I can go ahead and click Add, and where's that ClinVar data set? Did it finish downloading? Go back to here. There it is at the top of my list. Oh, I forgot. What's going on is that by default we're just looking at text files. This is a good opportunity to show you that we support importing bed files. You can curate your own reference genomes, FASTA with 2-bit, VCF of course, fixed wig, fixed step, GTF files for gene annotation sources, and if you want to just have any file extension and give it a try, we'll treat it as a limited file and do our best to import it as well. If we switch on over to VCF, we can see our latest downloaded file here. Select that, and this very quickly then shows us a preview of that VCF data. In this case, VCF has a couple mandatory fields like quality and filter. We can drop those, we don't need them. RS looks like a repeat of the identifier field, and same with RS position. This VP field is not that interpretable, but actually the rest of this looks very useful. You get to see things like the HGVS notation, etc. So we very interactively are able to see what we're looking at. Go ahead and import it. Again, it detects the right genome assembly. And this is another thing we do is we fill in the documentation for each field from that VCF file. So all these descriptions were in the header of the VCF file, and we also have a section where we capture that full header of the VCF file so we don't lose anything that might have been stashed in there that would have been useful from a provenance perspective. So we go ahead and hit Next, and let's convert this data source. And voila, we have a very quickly computed, it's actually computing a couple extra things. It's creating an index on the identifier, so I can search by RSID and pull up all of the variants in ClinVar, as well as being able to compute what we call the coverage information, so that we can very quickly zoom out and see information about it. So here's that ClinVar data set. Notice it doesn't really match the way we're writing our previous 2013 ClinVar data source. This shows that we actually, it's not a one-shot deal. If you want to edit an existing data source, you can just click on Edit here. Let's change the formatting here to match our existing formatting for this. All right, so 303 and hit Save. Now refresh, and there we go. So now is our new ClinVar data set, and we can plot this into our data, and now we can start to see where those ClinVar variants are compared to our samples. And then as I mentioned, you can actually search here and find any RSID in that ClinVar data set. It's automatically indexed that useful field. Okay, so plotting is great, and this is stuff you can do with Genome Browse, but you know, I have some variants here, and I want to know really what's going on with these variants compared to these data sources I just curated. So let's go ahead and annotate using our new annotation mechanism. So DNA-Seq, annotate and filter, and I'm going to add tracks here. As I mentioned, anything in this dialog is a usable track for annotation. I can grab some of these things that are on our server. If I don't want to download them, that's fine. I could also select things and download them based on our multi-threaded background downloader. And my user annotations folder contains my new ClinVar track and my DNase hypersensitivity site. So let's go ahead and select those. And there they are, and I'm going to annotate those one by one. So it asks me what I want to do. Do I want to filter and only look at sites or variants inside DNA hypersensitivities? No, I'm not going to filter. I just want to annotate. Similarly with dbSNP, let's go ahead and just annotate, not filter. Go ahead and continue on here. So now it's doing a quick pass of my 3,000 variants and looking at where they fall in those 1.3 million hypersensitivity sites detected by the ENCODE project, and it creates a report for me. And then it also very quickly annotated against ClinVar. I found that 19 of my variants were inside of, or 17 of my variants matched ClinVar. The other two that were matched or inside but not matching would have a different set of alleles. And also we found that about 800 of my variants are in DNA's hypersensitivity site. So that's very useful information. I can pull up these reports. And actually one thing I can show you that's new in SVS8 is, although we've always been able to count how many variants are in one category or another on any column, right-clicking on a column now allows you to pull up a quick pie chart, which is a useful way to sort of visually see, okay, that about, maybe a little more than a third and less than half of my variants are in DNA's hypersensitivity sites. So I think we've covered the ability to use both public and local annotation sources to empower your own annotations. Let's go ahead and touch on some of the other things that we've been working on. As I mentioned, this has been an infrastructure release, and we've been really just doing the things we've always wanted to do, such as rebuilding this data annotation library to be the best thing we can imagine. So one thing we'll touch on in a little bit in the next demo is that this is essentially an ability to categorize or organize your favorites locations. And so although we're looking at just these existing library locations, you can add your own locations to that list. It's sort of a favorites bookmarks type system. But one thing I really got to give a lot of props to, one of our engineers has been putting an immense amount of effort into this, is that we have essentially been hearing from our users that although they love Genome Browse and they want to use it in publications, just taking a screenshot is not exactly the ideal way to get that beautiful rendering into a context where you can place it into, a website or a paper. And so we've been taking in all of that feedback and spending a lot of time rethinking how we would export this, visualization system into all these different formats and contexts that you might want to use it. This has not been an easy effort, as you can imagine. Rendering to a screen is one thing. Rendering to a potential image with different, density of data, DPI with potentially different page layouts or a PDF can be quite complex, and we actually had to rethink and re-architecture about half of our entire layout system and our Genome Browse code base to enable this feature. But it's very exciting, and we actually have in SVS8 and in Genome Browse 2.0 the beginnings of our save as image functionality. So let's go ahead and look at what that will look like, or what that is looking like now. So I'm in my project here. This is another place where I can showcase that Genome Browse and SVS can now play very easily together. While we were plotting spreadsheet data before, at any time you can pull up a Genome Browse window that's essentially a node in your SVS project and just look at data from either your project by going to this project icon here, or maybe any directory. You can go to Browse, select a directory, as well as I mentioned, this library is here for you to customize. For example, let's go ahead and add a sort of a bookmarked location here. So if you have data on an external hard drive, I have this backup hard drive. This actually has my own exomes on it. So let's go ahead and pull this folder in there. You can actually recursively have folders under folders if you want to. So here's my family trio. I'm just going to place that under my local list of favorites. And let's go ahead and plot my own exome and my variants. And the cool thing is, notice it says I got two things selected here. You don't have to just hit plot now. You can just hit plot, but you could also go ahead and grab some more things that you might want to look at. Let's pull in our ClinVar database that we just selected and the human reference sequence as well. So go ahead and plot all of those things. So we get 4 or 5 different data sources in our plot, and we can start to navigate around ERCC6. It's an interesting gene. This gene actually has a mutation in my genome that I've always been curious about. It's a nice little splice site variant right beside the tail of this exon. So I could adjust, maybe if I want to take a picture of this, some of the settings to be able to very quickly to view this type of data. So here's a nice pile-up view. Not a lot going on with ClinVar. Maybe I'll go over here where there is some annotations. Well, let's see. Yeah, now we get a little bit of everything going on. Okay, so it's a nice little picture. Maybe I want to adjust the size of this, of where I'm at. And you can see the gene tracks, add a little bit more dimension to that. Okay, so let's go ahead and use that save as image capability to get a picture of this. So save as image, and there it is. I can actually change whether I want that full domain view to be on there. I do like that, so I want that in there. It gives us a context of where I am in the current chromosome, and you can see the renderings of all of those variants and pile-ups and genes, etc. So let's go ahead and save this as, you know what this really is it's a selfie. It's just a selfie of my exome. So I'm gonna, this is my selfie exome.png. So I didn't get a chance to watch the Oscars. I don't know, maybe some of you did, but from what I can tell, the whole point of the Oscars was to really elevate the selfie to a whole new level of awesomeness. So now that I got probably one of the few exome selfies going on, we can make our own little Twitter history here and see if we can get this exome selfie retweeted. So let's go ahead, and if you haven't followed me, I'm on Twitter at Gabe Informatics. So let's do alpha exome selfie, first 5 retweets, get a at Golden Helix Inc. t-shirt. That might prime the prompts. We can make our own retweet history in our own bioinformatic community here. Retweet me, hashtag. Yeah, someone already knows what I'm going for here. All right, so now I just need to attach my save as image. Here's my selfie. Go ahead, and I'll keep my phone open, and if we see this thing burning up, we'll come back to it and see how it does. All right, so just to give you a sense of how much fun you can have. Oh, I already got it retweeted. Oh my goodness, that is fast. You guys are good. All right, someone got a t-shirt. Nope, there it is again. I got a little buzz on my desk here. Okay, so lots more things going on in SVS 8 than we could possibly cover. One thing I did want to touch on is that people who are doing genome-wide association studies and doing that type of association testing between potentially common SNPs and complex traits are definitely not left in the lurch with this release. Not only do you get all those rich sets of annotations and visualizations, and we have actually continued to invest in bringing this platform to the place where we've always wanted it to be. Again, it's another infrastructure investment that we, in this release, brought back Haplotype Trend Regression. It was in our previous Helix Tree 6 product line and never had a chance to fully support it, and now we were able to bring it up to date with a modern architecture and modern feature set. So that includes the ability to correct for covariates and being able to create haplotype blocks or be able to create haplotypes based on pre-computed blocks. So let's look at what that looks like in SVS. This also gets us a chance to look at a different type of data. Let's go ahead and hit save and open up a project I have where we have, so this is a project that contains, and I already have filtered down here, a selected set of pruned SNPs. And it's a case control study, but we have a blood pressure phenotype for these 565 samples. Now in my project, I've already done some single marker tests, but let's say we want to explore the potential for there to be a haplotype-based signal here that we need to capture. So we go to our genotype menu. We always have these other types of genotype methods, such as genotype association testing. We're going to go to Haplotype Trend Regression. And so now I can use a set of pre-computed blocks or a set of moving windows. These blocks are essentially LD blocks. So these are run by our LD block detection algorithm, and it creates this output spreadsheet. And as you can see, I got about 34,000 blocks here. I select those, it detects those, and I'm going to use those blocks to compute haplotypes and then associate those haplotypes with this continuous phenotype here, this blood pressure. Let's go ahead and correct for gender as well. So I don't want that, do a full versus reduced model. And I can go ahead and let this run. As you can see, it can go through at a pretty quick click, and we would probably only have to wait another 60 seconds, but I'm going to go ahead and cancel it so that we can pull up the results spreadsheet and look at this data a little interactively as sort of our last thing we touch on here. All right, so here we go. This is a full-blown SVS output statistics spreadsheet. We have our information about our blocks, the number of haplotypes computed in each one, and our regression p-values. Just like anything in SVS, you can right-click on a column and get context-specific actions. So we can pull up this variable as a plotted data source in Genome Browse, get that nice full domain view. We're looking across their entire genome here, and I can grab this this property or this we'll call it our node in our plot viewer, or essentially this item, and change its styling to be based on chromosome to get that standard Manhattan plot. But remember, this is Genome Browse, so you can use your mouse wheel. I should point out you can still use that standard click and drag zoom mode if you prefer that. But now we get to see that there is a signal here, and let's pull in a little context to look at this signal. So by that I mean we're looking at the statistics output. We can, what I like to always do is kind of look at the data underlying the statistics. If I go to my add data sources, here's our data source library. I mentioned this project interface is here. You can plot other spreadsheets, any spreadsheet that contains genomic data. And we get to choose how we want to plot it. We can plot that as a variant rug like we were looking at before. Because this is a big set of 565 samples, we can build an LD estimations of that population group and plot that. So we get a nice density rug showing you where the data is. As we zoom in, we get to draw that data, and now we're drawing that LD information. And let's go ahead and add into this also, we can overlay the marker blocks that we were using in our statistic. So I can load in our LD item, there's an option to look at marker block information. We can go ahead and load those marker blocks, those 34,000, see where they fall. And these are essentially the blocks that were being used in that statistics. And you can see we're actually in this FTO gene. One thing we did with SVS, with our latest round of data curation, since our new file format is so generous in being able to easily compress stuff, is that we added more annotations to all of our sources. So if you click on FTO, for example, you know, why are we getting a strong significant signal here in FTO? With a blood pressure phenotype, if we scroll on down, it becomes extremely obvious that this is a gene that has been shown to have a strong association with body mass index, obesity risk, and type 2 diabetes. In fact, you know, it's sort of this fat mass gene. So it is a very cohesive story that we're getting a strong haplotype association signal for a blood pressure phenotype right in this gene here. So that covers our interactive demonstration of our new haplotype trend regression that we'll have in SVS8. And I wish I could have more time to cover everything else, because of course there are many, many other things that we've been continuing to improve. We will always be placing this in very vivid detail in our release notes that we'll have in our new version of our manual. But also for this release, we'll have a What's New in SVS8 page up on our website that you can go check out to capture some of these highlights and see if there's anything else that might be useful to your own research there in this latest release. And don't feel like you have to wonder about whether you should or can upgrade to this. You always can, if you're a customer of SVS, use the latest releases for free. It is something that we sort of include in our overall support contract. And of course, if you want to demo SVS, you can do that as well. Just go and fill out an evaluation request, and you'll have a fully featured version of the software that you can use on your own data or our own tutorial data to kind of do some things similar to what I've been showing you today. And with that, I'm going to turn it back over to Andreas, and we'll take as many questions as we have time for. And as I mentioned, we'll capture these questions, and if we want to, we can follow up via email.
All right, super. Gabe, this was an excellent overview. Thank you very much. By the way, I just retweeted your tweet, so I'm going to pick my t-shirt up in a second. Yeah. I need double X. No, in all seriousness, I think you and your team, you have worked very, very hard to push SVSA to this new level, and I think we're very excited to be able to show this to our audience. For those of you who join us for the first time, we actually do these events on a regular basis, about once a month, sometimes a little bit more often. This one was by nature, or by definition, very product focused, yeah, and you can sense the level of pride that we have for this particular release. Other webcasts are more about best practices. How do you do an NGS analysis or a study or a GWAS study, or we talk about specific research results. In fact, we just finished up an abstract competition. We will have over the course of the year about 6 customer presentations that talk about how they use SVS in the context of their particular research area. Now, with that, I would like to go through some of the questions that have been asked. And I have to apologize, we will not be able to cover all of them. But what we will do is we'll write up a little blog that will cover the most important ones so that you will get your information. So the first question is, I think, about something I mentioned about the adoption of SVS for educational purposes, and the desire to hear a little bit more about this. It's an interesting situation that we have now seen here in the US in particular, but probably is everywhere in the world the same. We have a lot of new people coming in this area. Students are wanting to learn more about this type of research and what it entails, and there's this huge hurdle to learn first scripting and deal with the technicalities of the public domain software packages that are out there. In particular, if you all you have is basically command line tools, big issues are visualization, then how to really effectively do annotations to specific data sets, and then just handling the massive amounts of data. So those are the challenges that the students are dealing with, and obviously the software, as you've seen, deals with this very easily, and so the threshold to enter the field has been lowered. We provide additional help. There's the site, as I mentioned earlier, has a plethora of videos and other materials that we can use for self-study purposes. We have tutorials that can be used for coursework, and then, quite frankly, here we at Golden Helix, we advise. Should you be a professor that has to put a course together, we advise on how to best organize a semester, and that basically makes easy work for everybody all the way around. So that's essentially the upshot here. We're provide a flexible license model. If that's something that's interesting to you, just reach out to us. The second question is, we just launched SVS8, and obviously there's a lot of new things, but there's a question on what are our plans with the platform going forward. Maybe can you share a little bit what kind of is on the docket for maybe this year?
Yeah, absolutely. So for this year, like any infrastructure release, it opens a lot of doors for taking any type of workflow to a whole new level. One thing we've noticed is that NGS has really started to come to the point where people can start to do very large-scale studies with NGS data. So we're going to spend a lot of time on our statistics side this year, expanding out our capabilities for large N analysis workflows. So that includes looking at new collapsing method algorithms and ways of which we do collapsing, other types of ways of sort of inferring association with complex diseases. On the more small end side, doing more targeted trios or single sample analysis, we have also great big plans. We really are thinking about how to rethink the total experience to simplify it for someone who wants to do this in a very repetitive or lockdown workflow. So that's what we're planning on doing, taking all this great infrastructure for being able to annotate against all these public data sources and making the annotation and filtering even easier.
Yeah, all right, super. The next question is, when is this release actually available?
Okay, so on that, we are looking at essentially the full finished version of SVS 8 today, but we need to kind of wrap it up with our documentation and standard testing process, so we'll be planning on getting it out the door late next week, so by the latest Friday of next week. You'll see it in your upgrade window if you are an SVS customer running SVS 7 or etc. You'll see an update available, or after we ship, you'll be able to, it'll be the default download if you're downloading a new copy.
Mm-hmm. Yeah, okay. There's a question from someone in India about hardware requirements. Has they changed from 7 to 8, or maybe in general the question is, what are they?
Yeah, they really haven't, and in fact, things are almost easier with the new TSF file format in the sense that things can just run very efficiently. But the hardware requirements are wholly dependent on sort of the scale of data you're working with. If you are doing with whole genome data, you generally need more RAM simply because there's a lot of moving parts. We can still efficiently run, though, on a workstation. So, even a workstation with 16 gigabytes of RAM is sufficient. But maybe you want a little bit more for doing whole genome data. If you're working with exome data or SNP arrays, you actually can run that on your laptop, or pretty much any computer built in the last 3 or 4 years will be sufficient, and it's just really a matter of how much horsepower you want to throw at it. The average, I think, minimum requirements that we suggest is 2 gigabytes, probably safely 4 gigabytes of RAM, and a couple CPU cores.
Yes, I see here on my question window that questions are still plowing in, and as I said to you, we'll get to those. I think we can maybe do 2 quick ones here. The next one here for you, Gabe, is there a functionality for plant data sets?
Oh, absolutely. As you might have seen, there was although we were looking at human data sets in our library here, let me quickly look at our, say, public annotation server. If you switch this on over to any assembly, we have data in very many different species, already curated, as well as our team is happy to help you bring in a new reference sequence from any number of plant and animal species. And the statistics and the annotation and filtering mechanisms are all going to support non-human data.
Mm-hmm. Okay, super. And then the last question that we have time for today is, apparently this is the question is, if I work in a project with collaborators outside of my university, how do I give them access to the software? That's more of a licensing question. Actually, your collaborators would have to buy the license as well at some point. We are very flexible to give them initial access to that, so that they can test out and make sure that they're comfortable with that choice. So we run this problem all the time. If that happens in your project, just feel free to reach out to your territory manager here at Golden Helix, and then we will arrange for a trial license to be issued to that collaborator so they can start that process. Again, lots more questions. We'll take care of those in a separate blog, and I really appreciate the huge attendance here. This was a very successful and a webcast from our perspective. Thank you very much for joining us, and we will be in touch with more information on SVS. Thank you very much.
Thank you, guys.